بسم الله الرحمن الرحيم. طلبة المرحلة الثانية نبدأ بالمحاضرة السابعة بشرح توضيحي للمحاضرة السابعة لمحاضرات الدهون. This lecture include cholesterol synthesis and utilization. The synthesis and utilization of cholesterol must be tightly regulated in order to prevent overaccumulation and abnormal deposition within the body. Of particular clinical importance in the abnormal deposition of cholesterol and cholesterol-rich lipoprotein in the coronary arteries. Such deposition, eventually leading to atherosclerosis, is the leading contributory factor in the disease of coronary arteries. إذن اللي يهمنا بالclinical importance اللي عنده clinical importance هو abnormal deposition of cholesterol، ترسب الكوليسترول بشكل غير طبيعي، وأيضا الكوليسترول rich lipoprotein مثل عندنا ال LDL in the coronary arteries. هذا هو اللي يهمنا من الناحية السريرية. Such deposition مثل هذا الترسبات اللي تصير بالكوليسترول lead to atherosclerosis in the leading contributory factor, which is the leading contributory factor in disease of coronary arteries. Structural biochemistry of cholesterol. Cholesterol is a 27 carbon compound consisting of 4 linked hydrocarbon rings forming the bulky steroid structure. إذن الكوليسترول مكون من 27 كربون atoms compound، يتكون من 4 linked hydrocarbon rings، 4 حلقات من الهيدروكربون rings which forming the bulky steroid structure. There is a hydrocarbon tail linked to one end of steroid and a hydroxyl group linked to the other end. إذن في أحد نهايات هذه الحلقات أحد النهايات مرتبطة بالهيدروكربون تيل والنهاية الأخرى بالهيدروكسيل جروب. Cholesterol is known as sterol because it is made out of alcohol and steroid. ليش نسميه ستيرول؟ لأنه مكون من الكحول مع الستيرويد. And this is the structure of cholesterol. قلنا يتكون من 4 carbon ring مرتبطة مع بعضها، ومن one end of the steroid بينه الهيدروكسيل group and the other end is hydrocarbon group. Note أن ملاحظة أنه الأسيتايل-CoA utilized for cholesterol biosynthesis is derived from an oxidation reaction. Example fatty acid or pyruvate. إذن الأسيتايل-CoA اللي نستخدمه في بناء الكوليسترول هو مصدره من الأوكسيديشن reaction، إما الفاتي أسيد oxidation أو من الأوكسيديشن of the pyruvate in the mitochondria and is transported to the cytoplasm. يعني الاثنين الأوكسيديشن occur in the mitochondria, then the acetyl-CoA is transported to the cytoplasm. Acetyl-CoA can also be synthesized from cytosolic acetate derived from cytoplasmic oxidation of the ethanol, which is initiated by the cytoplasmic alcohol dehydrogenase. إذن other source of acetyl-CoA هو من cytosolic acetate derived from cytoplasmic oxidation of ethanol. All the reduction reactions of cholesterol biosynthesis use NADPH as a cofactor. إذا نتذكر أن الفاتي acid synthesis كان أيضا نحتاج إلى NADPH as a reduction reaction. بكل التفاعلات الريدكشن اللي كانت بالفاتي acid synthesis احتجنا إلى NADPH. فأيضا نفس الشيء بالنسبة للكوليسترول بالريدكشن reaction نحتاج إلى NADPH as a cofactor. Now cholesterol synthesis. The process of cholesterol synthesis can be considered to be composed of 5 major steps. إذن 5 major steps for cholesterol synthesis, where the reaction that culminate in the synthesis of isopentenyl pyrophosphate and its isomeric form dimethyl allyl pyrophosphate are commonly referred as mevalonate pathway. Now in the first step, acetyl-CoA are converted to 3-hydroxy 3-methylglutaryl-CoA. Second, hydroxymethylglutaryl-CoA is converted to mevalonate. Third, mevalonate is converted to the isoprene-based molecule, which is isopentenyl pyrophosphate. Fourth, isopentenyl pyrophosphate molecule are converted to squalene, and fifth, squalene is converted to cholesterol. And so these are the 5 steps of cholesterol synthesis. قلنا الخطوة اللي ينتج عنها تكوين الميفالونات نسميها الميفالونات pathway. First, the biosynthesis of mevalonate. Initially, 2 molecules من الأسيتايل-CoA condense to form acetoacetyl-CoA, catalyzed by cytoplasmic thiolase. إذن هاي الخطوة تشبه الفورميشن أو السينثيسيس مال ketone bodies. لمن 2 molecule من الأسيتايل-CoA condense and form acetoacetyl-CoA, and also catalyzed قلنا by thiolase enzyme. Acetoacetyl-CoA condenses with further molecule of acetyl-CoA. Then another molecule of acetyl-CoA will be added to the acetoacetyl-CoA and catalyzed by hydroxymethylglutaryl-CoA synthase enzyme to form hydroxymethylCoA. إذن acetoacetyl-CoA condense مع another molecule من الأسيتايل-CoA to form hydroxymethylglutaryl-CoA. Then hydroxymethylglutaryl-CoA is reduced to mevalonate by NADPH. يصير بينه reduction in the presence of NADPH in a reaction catalyzed by reductase enzyme, which is hydroxymethylglutaryl-CoA reductase. This last step is the principal regulatory step. إذن هذه الخطوة هي formation of formation of mevalonate is the regulatory step in the pathway of cholesterol synthesis and is the site of the action of most of effective class of cholesterol lowering drugs. معظم الكوليسترول lowering drugs تشتغل على هذه الخطوة، مثل الستاتين. عندنا drugs اللي يعمل lowering الكوليسترول، عندنا مثال علينا الستاتين which are hydroxymethylglutaryl-CoA reductase inhibitor. إذا لما يعمل inhibitor hydroxymethylglutaryl-CoA reductase enzyme راح نمنع formation of mevalonate from the hydroxymethylglutaryl-CoA. إذا الستاتين هو inhibitors للreductase enzyme، هو أحد أنواع الدراكس اللي تستخدم for lowering the cholesterol level. And as we say, this picture show the formation of mevalonate. As we said, 2 molecules من الأسيتايل-CoA condense in the presence of thiolase enzyme to form acetoacetyl-CoA, and in the presence of synthase enzyme another molecule of acetyl-CoA will be added to the acetoacetyl-CoA or condense with the acetoacetyl-CoA and form 3-hydroxy-3-methylglutaryl-CoA. Then the 3-hydroxy-3-methylglutaryl-CoA will undergo reduction to form mevalonate, and this steps need for the enzyme reductase, which is hydroxymethylglutaryl-CoA reductase, and this is the regulatory steps. This step is a regulatory step. As we said, the drugs lowering cholesterol, drugs that lower the cholesterol act on these steps. They act on the reductase and act as inhibitor for reductase enzyme, such as statin drug. Now the formation of isoprenoid units, which is isopentenyl pyrophosphate unit. Mevalonate is finally converted to isopentenyl pyrophosphate by 3 phosphorylation steps and 1 decarboxylation steps, and these required ATP. إذا يتحول إلى الميفالونات إلى الأيزوبنتانيل pyrophosphate ب 3 steps of phosphorylation and 1 steps من decarboxylation. And this picture shows the formation of isopentenyl pyrophosphate from mevalonate. وقلنا تحتاج إلى 3 steps من الphosphorylation and 1 steps من decarboxylation. isopentenyl diphosphate or pyrophosphate هي معنى البايرو معناها داي يعني بيها 2 phosphate group. إذن isopentenyl diphosphate is isomerized by shift of double bond to form حتى يكون لنا الايزومر. احنا قلنا الايزوبنتانيل بايروفوسفيت بينه ايزومر اللي هو الداي ميثايل الايل دايفوسفيت. اذا الخطوه اللي بعد تكوين الايزوبنتانيل دايفوسفيت يصير بيها ايزومرايزيشن يكون لنا الايزومر of it which is dimethyl allyl diphosphate or pyrophosphate, and then this will condense with another molecule of isopentenyl diphosphate to form 10 carbon intermediate which is called geranyl diphosphate. Further condensation with other molecule من isopentenyl diphosphate to form farnesyl diphosphate. So 3 molecules of isopentenyl pyrophosphate condense to form farnesyl pyrophosphate. إذن 3 molecules من isopentenyl. The first molecule مع the second molecule condense to form geranyl diphosphate, then another molecule من isopentenyl pyrophosphate condense with it to form farnesyl pyrophosphate. فبالنتيجة نحتاج إلى 3 molecule of isopentenyl pyrophosphate to be condensed and form farnesyl pyrophosphate. And this show how farnesyl pyrophosphate is formed from condensation of a 3 molecule of isopentenyl pyrophosphate. 2 molecule of farnesyl pyrophosphate then condense to form squalene by the action of squalene synthase in the endoplasmic reticulum. إذن the formation of squalene يحتاج إلى 2 molecule of farnesyl to be condensed, farnesyl pyrophosphate to be condensed to form this squalene. And this the formation of squalene to farnesyl pyrophosphate with the presence of NADPH give the squalene. Now oxidosqualene cyclase then cyclize the squalene to form lanosterol. إذن الإنزيم اللي هو oxidosqualene cyclase then will cyclize the squalene to form lanosterol. Finally, lanosterol converted to cholesterol through 19 steps process. إذا يحتاج إلى 19 steps حتى يتحول اللانوستيرول إلى الكوليسترول. And this is how it will convert it to cholesterol. طبعا المعادلات ما مطلوبة منكم. All body cells manufacture cholesterol for both membrane structure and other uses, with relative production rates varying by cell types and organ function. إذا جميع أنواع الخلايا البودي can form cholesterol which is needed for membrane structure and other uses، لكن تختلف نسبة تكوين هذا الكوليسترول من خلية لأخرى ومن حسب الأورجانز أيضا حسب الأورجان function. 80% of total daily cholesterol production occur in the liver. إذا أعلى نسبة من تكوين الكوليسترول تكون in the liver. Other site of higher synthesis rates include the intestines, adrenal glands and reproductive organs. The cellular supply of cholesterol is maintained at a steady level by 3 distinct mechanisms. First, regulation of 3-hydroxy-3-methylglutaryl-CoA reductase activity and levels. Second, regulation of excess intracellular free cholesterol. Third, regulation of a plasma cholesterol level via LDL receptor-mediated uptake and HDL-mediated reverse transport. Even by these 3 mechanisms we can regulate the level of cholesterol and maintain it at a steady level. The utilization of cholesterol. Cholesterol is transported in plasma predominantly as cholesterol esters associated with lipoproteins. إذا الناقل للكوليسترول هو اللايبوبروتين، ينتقل بالبلازما بشكل الكوليسترول استر بواسطة اللايبوبروتين. الديتري cholesterol is transported from the small intestine to the liver within chylomicron. إذا الكايلوميكرون هو اللي ينقل لنا الكوليسترول الدايتري cholesterol from the intestine to the liver. Cholesterol synthesized by the liver as well as any dietary cholesterol in the liver that exceed hepatic needed, it is transported in the serum within LDL cholesterol. إذا الكوليسترول اللي ما يحتاج اللي يصنعه الليفر أو اللي يجي من dietary cholesterol والليفر not need for it أو exceed the need for the liver is transported by LDL lipoprotein or low density lipoproteins. Cholesterol is used to produce first the steroid hormones required for the normal development and functioning. For example of these steroid hormone is sex hormones like estrogen and progesterone in women and testosterone in men. These hormones trigger the development of the physical trait characteristic of adults, women and men. They also play a role in reproduction. Other steroid hormones produced from cholesterol include cortisol, which is involved in regulatory blood sugar level and defending the body against infection, and aldosterone, which is important for retaining salt and water in the body. The body can even use cholesterol to make a significant amount of vitamin D, the vitamin responsible for strong bones and teeth and when the skin is exposed to sunlight. واحنا قلنا تتذكروها إنه الفيتامين دي قلنا synthesized endogenously from 7 dehydrocholesterol اللي موجود بالسكن after exposure to sunlight. Cholesterol is also used to make bile, a greenish fluid that is produced by the liver and stored in the gallbladder. The body needs bile to digest food and that contain fat. Once the fat is digested, bile helps the body to absorb it. Then bile acid is important for digestion and absorption of fat. The presence of bile in the intestines is required before cholesterol can be absorbed from the food. The body also needs bile in order to absorb vitamins like vitamin A, D, E and K as we said لأنه هي fat soluble vitamins فأكيد راح يحتاجون البايل أيضا حتى يصير بهم absorption from food or from supplement. سواء كان مصدر الفيتامينز من الفود أو supplements يحتاجون إلى bile for their absorption. The body has the ability to make all cholesterol it needs for these various functions. A diet that contains animal products, however, also supply cholesterol to the body. إذن هو body can synthesize all its need of cholesterol. لكن أيضا الدايت اللي هو الانيمال برودكت أيضا تحوي على الكوليسترول وممكن أن تجهز البادي بهذا الكوليسترول. In an effort to balance these two sources of cholesterol, the body adjusts the amount it produces each day. احنا قلنا السورس من الكوليسترول كان be synthesized endogenously والبادي كان synthesize all its need of the cholesterol. أيضا قلنا الكوليسترول ممكن أن نحصل عليه من الدايت فلازم يكون أكو توازن بين هذين المصدرين من الكوليسترول عشان يحدث هذا التوازن. For example, if you eat many food from animal source, your body gets substantial dose of cholesterol from the diet called dietary cholesterol, so your body then slows down its own production of cholesterol. يعني إذا كان الفود يحوي على نسبة عالية من الكوليسترول فإذا البادي يقلل ال formation of cholesterol ويقلل ال production of endogenous cholesterol. On the other hand, when most of food you eat come from plant source ما بيها الكوليسترول, your body manufactures more cholesterol in order to meet its need. يعني بهالحالة هاي يزيد الفورميشن, البادي يزيد الفورميشن of cholesterol إلى المستوى اللي يحتاجه. فإذا هو البادي يعمل بالانس بين الاندوجينوس كوليسترول اللي يكونه البادي مع الدايتري كوليسترول. إذا زادت كمية الدايتري كوليسترول اللي الشخص يتناولها فالبادي يقلل السينثيسيس اوف كوليسترول, وبالعكس إذا قلت كمية الكوليسترول المتناولة from diet يعني بهالحالة هاي يزيد الاندوجينوس كوليسترول synthesis. The body can also eliminate some excess cholesterol through bile. Whenever bile is released into the intestine and a portion of it is absorbed back to the body to be used again. The remaining bile is excreted in the feces, and to help maintain the cholesterol balance, the body can dissolve excess cholesterol in the bile and can also convert more cholesterol into a bile acid so that cholesterol will be excreted with feces. إذن أيضا ممكن البادي يعمل excretion لهذا excess cholesterol أو eliminate this excess cholesterol through bile, either by formation more amount of bile acid or by dissolving the cholesterol in bile and to be excreted with feces. Now in summary, cholesterol is a lipid with unique structure consisting of 4 linked hydrocarbon rings forming the bulky steroid structure. Second, the acetyl-CoA utilized for cholesterol biosynthesis. Third, all the reduction reaction of cholesterol biosynthesis is needed for NADPH as a cofactor. For about 80% of total daily cholesterol production occur in the liver. And fifth, cholesterol is transported in the plasma predominantly as cholesterol esters associated with lipoprotein. Thank you very much.